Dear students, in the last class we have already learned or we have already discussed about body fluids, what are body fluids, the importance of body fluids, and also we have discussed about the blood plasma. In this lesson, we are going to discuss about the formed elements. Now, when we are talking about formed elements, in many textbooks, you will find that formed elements are also known as blood corpuscles. Do you know what is the meaning of this blood corpuscles? Why the special name is given? In many textbooks, you will also find it is written as blood cells. So you know what is the meaning of blood cells. But do you know what is the meaning of blood corpuscles? This special name is given because it is a special name for a cell. It is a special type of cell where the cells are suspended in a liquid medium. That means when a cell is suspended, especially in a liquid medium, then we will often define it as a corpuscle. Fine. This formed elements or blood corpuscles, they constitute about 45% of the blood. We have already discussed this in the last class, right? Now, these formed elements are of three types. You already know from class 9 onwards, you already know about this. What are they? Red blood corpuscles, white blood corpuscles, and platelets. You already know about this. Now, these red blood corpuscles, they, out of 45%, they constitute about 92.4%. 92.4%. All these are approximation. It might vary to some extent from textbook to textbook. So don't worry about that. WBC is around 0.15% and platelets are nearly about 7.45%. Okay? Now, when we are talking about the RBC or the red blood corpuscles, we all know that red blood corpuscles are also known as erythrocytes. Isn't it? They are also known as erythrocytes. And the main characteristics of these erythrocytes is non-nucleated, non-nucleated, oval, okay, or and it is biconcave. Meaning, what do you know the meaning of biconcave? It is the RBC is not perfectly oval in shape, but somewhat like this. Fine? That means the two surfaces here is curved somewhat oxygen. Okay, so it contains a pigment hemoglobin that carries oxygen. Now, you need to know the how much amount of RBC is present in case of males and in case of females. In case of males, it is around 5 million per millimeter cube. And in case of females, it is around 4.5. Fine? Now, let me talk about the functions of RBC. When we are talking about the functions of RBC, the first thing that you have already learned or you already know that it helps in exchange of gases, right? It helps in exchange of gases. Which gases? Gas, oxygen and carbon dioxide. So it helps in exchange of gases like oxygen and carbon dioxide, oxygen and carbon dioxide between tissues and lungs. Fine. The main function here is it helps in exchange of gases like oxygen and carbon dioxide between the tissues and lungs. Apart from this, it also maintains the alkalinity of the blood, that is 7.4 pH. We have already learned in the last class. Also, it helps in the formation of bile pigments, stool, or urine formation by breaking down of the hemoglobin of RBC. What are those pigments? The pigments which I mentioned right now. What did I say? It helps in the formation of bile pigments, urine, stool, like sarcogeny, uroglin, bilirubin, sorry, bilirubin. Okay, so these are the formation of bile pigments that occurs with the breakdown of hemoglobin of RBC. Now, coming to the next characteristics. What is it? The formation and maturation of this RBC is also known as chemopoiesis. What did I say? Formation and maturation of this RBC is termed as erythropoiesis. Right? So formation and maturation of RBC is known as erythropoiesis. Next, you need to know that when as you have already learned that the amount, how much amount should be there in RBC normally in your body. Now, if it is abnormal cases, like if there is higher count of RBC, then we refer this as polycythemia. Polycythemia. When there is lower count of RBC, then it is said to be is granulocytes. Another one is granulocytes, right? Granulocytes and granulocytes. Granulocytes is around 70%, okay, and granulocytes is around 30%. Now, when we are talking about granulocytes means it has got granules. That means there is presence of granules in the cytoplasm. Right, students? Presence of granules in the cytoplasm. Also, here the nucleus is lobulated. Here the nucleus is lobulated. Here we are going to have a detailed information just after this. Okay. Let me explain the terms first, then we are going to classify them further. Granulocytes. Means there is no granules present in the cytoplasm. Alright? Like that of the granulocytes. That means here there is absence of granulocytes in the cytoplasm. And here the nucleus is also not lobulated. Nucleus not lobulated. Okay? So from here only you can write down the differences between granulocytes and agranulocytes. Right? Now let's talk about the granulocytes by further classifying it. in detoxification. Okay. Also, what it does, it prevents from any kind of allergic reactions. Right, students? So, eosinophil functions in detoxification. Also, it prevents from any kind of allergic reactions. And its lifespan is around 8 to 12 days. Okay, its lifespan is around 8 to 12 days. On the other hand, basophil, its lifespan is around 12 to 15 days. Okay, and here neutrophil, its lifespan is around 2 to 4 days. Now, when we are talking about basophil, let me rub off the characteristics of eosinophil. Otherwise, you will get confused. Here, in basophils, as I said, its lifespan is 12 to 15 days, and here the nucleus is 2 to 4 lobe. That means here the nucleus is somewhat like this. Did you get my point? So nucleus is somewhat 2 to 4 lobe, and the main function here in basophil, that is, it contains heparin. It contains heparin. Very important function. What is heparin? What is its function? It prevents from blood coagulation. We are going to talk about blood coagulation in the next class. Okay. So just remember this, that it contains heparin, which actually prevents from blood coagulation. Right? Now, next coming to the neutrophil. Neutrophil, as I said, its lifespan is around 2 to 4 days. And here, the neutrophils, they are 2 to 7 lobe. Okay, it might vary 2 to 7 lobe. Okay, like this 2 to 7 lobe, it can vary from 2 to 7 lobe. And its main function is to engulf what? Engulf foreign substances. Let me write down. Its main function is to engulf foreign substances, writing it short, foreign substances by the process of phagocytosis. Now, what is this phagocytosis? Let me discuss phagocytosis first. What happens in phagocytosis? We have already have studied phagocytosis many a times before. But do you really know what is phagocytosis? Actually, what happens? Suppose a foreign substance. Okay, suppose a bacteria. This is a bacteria, suppose. This bacteria is entering into your body, and the neutrophil, what it does, suppose this is a neutrophil, and this is approaching towards this foreign substance. Fine? When this neutrophil, suppose this is a neutrophil, and this is the bacteria, okay? Or any kind of foreign substance. This is a bacteria. What happens? Neutrophil approaches towards this foreign substance and try to stick to the bacteria. So next step, what happens? tries to engulf that bacteria by the process of phagocytosis. How it happens? Just a minute. Let me rub. The bacteria, it engulfs the bacteria forming a vacuole like this. And the bacteria is present here. So this neutrophil, this forms a vacuole, and this vacuole is known as phagosome. Okay? So first of all, what it does, when the bacteria enters into your body, neutrophil comes in or approaches to the bacteria, then it tries to digest the bacteria by forming a vacuole, which is forming a vacuole that is the phagosome. And then what it does, in the next step, this there is a lysosome. This is a lysosome. This lysosome gets in touch with this bacteria, and what lysosome does, you already know that lysosome is always act as a suicidal bag, and this lysosome will try to digest this bacteria. Okay, digest this bacteria, and what will happen? It will suicide. It will commit suicide, and thus this bacteria will get destroyed. Okay, the bacteria will get destroyed.